My name's Chris Sassetti. I'm an associate professor at the University of Massachusetts Medical School, and my lab studies a disease called tuberculosis. Tuberculosis is one of the most devastating diseases, infectious diseases in the world right now. It affects tens of millions of people every year and kills 2 million of them. And really the challenges to controlling this disease are understanding who has the infection, who's going to actually develop disease from the infection, because only a small proportion of people that are infected actually develop disease, and we don't know why that is, and treating the people that have the infection. Unlike other bacterial infections that we can treat with antibiotics relatively quickly, the course of therapy that's required to eradicate a tuberculosis infection requires 6 to 9 months of multiple antibiotic therapy, which is really difficult or impossible to deliver throughout most of the world. Controlling this disease is really going to require a more fundamental understanding of both the causative agent, Mycobacterium tuberculosis, and the disease that it causes. So we're interested in understanding the fundamental physiology of this infection. My lab is interested in understanding how this organism adapts to different environments it encounters in the host. And that gives us an idea of what bacterial functions are really important for survival in that environment, and therefore identifies targets for new classes of antimicrobial compounds that we could use for treatment. Probably the most fundamental challenge facing any pathogen that infects our bodies is acquiring nutrients in that unusual environment. We've been studying how Mycobacterium tuberculosis acquires nutrients while it lives inside of our cells. And we've discovered that a really essential nutrient for growth in that niche is cholesterol. This bacterium has the unusual ability to degrade that compound, and an ability that we ourselves don't have, as a source of energy. And in defining that ability, we've, I think, defined a number of new and really interesting targets for antimicrobial compounds. Another challenge in tuberculosis is understanding actually who's going to get disease. It's thought that something like 1/3 of the world has been exposed to Mycobacterium tuberculosis, but only a small fraction of those people are going to develop disease. And in order to use our limited resources more wisely to control the infection, we'd like to understand what actually determines disease progression, what determines who's going to get sick, and hopefully in that way to be able to develop diagnostics that allow us to identify people who are at risk and allow those to be treated.